Hello everyone, in this video we're going to talk about cancer. Cancer, as you know, is when the gene regulation system goes wrong and there's a lot of mutations that are going on. So this is actually related to what we were talking about earlier, the differentiation of cells and stem cells. Here are some vocabulary that we know. The first one is called proto-oncogene. There's the word gene in there, so we are talking about a gene. Remember, what is a gene? So proto-oncogene is a normal version of the cancer-causing gene, right? So a lot of the times there will be a gene, let's say it's messing up with the cell cycle control or the cell cycle checkpoint. And sometimes the cell cycle is supposed to check for the DNA damage and allow the cell cycle to go into the G0 phase and fix the DNA damage instead of going on to the G1S, G2M, this whole cycle to allow cell division. But if you were to have the gene responsible for the G1 checkpoint that's mutated, before it's mutated, you will call it the proto-oncogene. And then once it's mutated, you would call it the oncogene because now it's causing cancer. In this scenario, it's causing cancer because it's not stopping the cell cycle when it's supposed to stop the cell cycle, and then our cell keeps on dividing even though there's a problem. So proto-oncogene and oncogene. And there are many, many, many different types of these genes, and there's many more that we haven't discovered yet. An oncogene arrives from a genetic change leading to an increase in either the amount of activity of the protein. Wait, this doesn't make any sense. So what it means is that so we have we're either increasing the amount of activity of the protein produced by the gene, or we're increasing the gene expression or the production of the protein. So if that makes sense, we have gene producing proteins, right? Either we're increasing the amount of proteins that are produced, or we are increasing the activity of the protein. So the amount isn't changing, but the activity raises. So that's how usually an oncogene works and how it will cause cancer. So let's look more into it. Okay, so there are three major ways that a gene can become, or a proto-oncogene can become an oncogene. Okay, so there's a lot going on right here, but let's focus on this part, this left part of the graph. We have proto-oncogene, but there's a translocation or a transposition that puts this proto-oncogene with a new promoter. So what that means is that if we were to have a DNA right here, right, and we have a promoter right here that's really active, it's constantly transcribing this piece of DNA right here, these genes. However, we have this proto-oncogene right here normally is regulated just fine. It's not doing a whole lot of transcription. It only does the transcription when it needs to. However, there's an event that can happen within your DNA that makes this gene jump over to this other side, or the part of the genome, right? Or we could even have two pieces of DNA, and a part of this DNA can jump over to this other part of DNA. So this event, this jumping of the DNA or jumping of the gene, could either be called translocation or transposition. We'll look at the difference between those two on the next slide. But either way, we're moving genes from one place to another place within the genome. If this movement of the gene puts this gene downstream of a new promoter, and this promoter happens to be really, really active, then you're going to have an increased amount of the gene product, an increased amount of this protein produced, right? So if you read this, movement of the oncogene to a position near an active promoter or a really active promoter, which means active promoter means it's on, right? It's on all the time, which may increase transcription, and the result is you're going to have a lot of these proteins. And if this protein were to be growth stimulating, a growth factor, for example, it's going to cause a lot of cell division. If you have a lot of cell division when it's not supposed to, these cells are going to become cancerous because this doesn't stop dividing, right? That's how cancer works. The second possibility is that a proto-oncogene can have something called gene amplification. So right here we have this one piece of gene. Normally, we might only have one copy of this gene. However, this is a type of mutation that just it can happen in your body, right? That this is also a mutation. We could have more copies of the same gene, right? So even though it's still, these 3 genes are still controlled by the same promoter or whatever, but now that we have multiple copies of this same gene, we're also going to have an increased amount of proteins produced. So this right here, amplification, increasing the number of copies of a proto-oncogene. The result is you get more of the proteins produced. If these proteins are in charge of cell division, there's going to be way more cell division than it's supposed to. The cells become cancerous. Now, last one is point mutation in the proto-oncogene or its control elements causing an increase in gene expression. So what does that mean? We have our proto-oncogene right here, just regular gene doing its job, everything is fine. There's a possibility that there could be a point mutation within the gene that makes, within a control element, this one's right here. So let's say this is a promoter or an enhancer element for gene. So if you remember, way back 2 weeks ago, we talked about gene regulation, right? There's promoter, there's regulatory gene and regulatory sequence. So if there is a mutation within the regulatory sequence or regulatory gene, there's a possibility that these regulatory gene or regulatory sequence supposed to be off most of the time and controlled, but this mutation is causing this regulatory sequence or gene to be on all the time. So now the result is, instead of having this production of the transcription, translation, the production of the protein controlled, now it's no longer controlled. We're having a lot more proteins being made even when it's not supposed to be made. Okay. Another possibility with this point mutation situation is that we can have a point mutation within the gene itself that's causing this gene to actually become an oncogene. So you can see the differences here. So what's the problem with this right here? This protein that's formed by this oncogene could be hyperactive and become, for example, degradation resistant proteins. So, when we talk about post-translational modification, sometimes the proteins need to be broken up when we don't need it anymore, right? So let's say this is a growth factor. We don't need the growth factors anymore, so we need to get rid of it. But this growth factor right here might be resistant to that. So then now we just keep on having a lot of growth factors and they're not getting destroyed when it needs to be. And now cells divide a lot, then we have cancer. Or we don't, hopefully. The first 3 types, this one, and this one, we all have an increased production of the certain protein. And this right here, we have an overly active type of that protein. So this right here shows you the difference between translocation and transposition. So translocation happens between 2 chromosomes where a part of the chromosome is going to switch, right? So this right here, as you can see, we have 2 chromosomes right here. They're not homologous chromosomes, A and B. And then they switch, which means, this part of these genes right here could be put downstream from a very active promoter, and now we're making a lot of proteins from this B when it's not supposed to. And here we have transposition, where instead of switching a part of the chromosome, a part of the DNA will actually kind of come up. It like, boop, jumps up, and then it gets inserted into another part of the genome, like this. And there's something that does this. It's called transposase. That's the enzyme that does this cutting and transposition. And this right here could be called the transposon. So here's another word for you. This is a type of gene called tumor suppressor gene. You might have heard it before, right? This name makes sense. Tumor suppressor gene. This is some sort of gene that regulates the genome. And what does it do? It helps preventing uncontrolled cell growth. It helps preventing a tumor from growing. So there could be mutations that can happen to the tumor suppressor genes. If the mutation decreases the protein product of the tumor suppressor gene, that can contribute to cancer onset, right? So you can kind of think of tumor suppressor genes as, let's say, the guards at a football game, right? They're supposed to throw out the people who are, drunk and disgusting and keeping the whole football game going just fine. So these are the tumor suppressor genes. These are the police. But if you have a mutation that decreases the amount of police in those GARs and tumor suppressor genes, or the proteins that are made from the tumor suppressor genes, then your cells are not regulated enough as is supposed to. Now your cell division is going to, keep on happening uncontrollably and cancer. So tumor suppressor proteins that are produced by the tumor suppressor genes, what do they do? Here's the main things that they do. The first one is called repair damaged DNA. So if you think about DNA, right? DNA, when there's a damage, either just caused by mutation on its own, which happens, or caused by radiations or whatever other environmental factors, a virus, there's a mutation in DNA, either way, there's a damage. Tumor suppressor gene can fix that damage so that your cell doesn't become cancerous, because cancer is a result of accumulation of mutations. But if we can fix it ahead of time, then the cell is not going to be cancerous. Tumor suppressor gene can also control cell adhesion to each other or to the extracellular matrix. So if you remember back in the days, you learned about if you're growing cells in a petri dish, they're only supposed to grow one layer. They only, the cells should not be clumped together and grow on top of each other all that often. That's because that's what cells do. That's how cells know whether there's a blank right here, we need to actually grow more cells, or if everything is fine, all the cells are there, we don't need to grow more cells. But if we were to grow cancer cells in a petri dish, then the cells will actually pile up instead of only growing in one layer. So tumor suppressor genes control or prevent cells from growing on top of each other. But if the tumor suppressor gene is damaged, which the tumor suppressor protein is damaged, or we don't have enough of it, then the cells can grow on top of each other. Then now we have uncontrolled cell division. The last one, tumor suppressor genes or tumor suppressor proteins can also inhibit the cell cycle. So if you think about each one on one of the cell cycle checkpoint, it's supposed to prevent the cell. First off, the cells need to check and make sure that the cells need to make sure that they have the right signal in order to divide, right? Is it time to divide yet? Is there growth factor? Is there enough nutrients? So the cells cannot divide if they shouldn't be dividing, and the tumor suppressor proteins are in charge of that. The tumor suppressor proteins can also inhibit the cell cycle if the cell is not ready, or so the cell, if the cell is not ready, or if the cell isn't receiving a signal to divide, then the cell will not divide, and tumor suppressor proteins are in charge of that. All right, so here's another type of gene called the ROS gene. The ROS gene makes the ROS protein. So whatever gene it is going to make that protein. So ROS gene makes a ROS protein that's a G protein that relays signal from a growth factor. So if you remember when we talk about the signal transduction pathway, right? There's a relay. There are relay molecules after the cell receives a signal. So this is an example of how cancer can be caused, okay? So if you have a mutation in the ROS gene or any pathway components within this relay pathway, these relay proteins, this could result in extra cell division. And 30% of the human cancer is actually affected by a mutation within the ROS gene. And if you don't remember, a kinase is transfers a phosphate group from an ATP to another molecule. So when we talk about the signal transduction pathway, a lot of those molecules are kinases. They transfer phosphates onto another molecule and then onto another molecule to activate them and allow the pathway to go on. So let's look at pictures of how this actually works. Oops. So on the top, this top picture shows you how the ROS gene is supposed to function. So if you can remember, cell-to-cell signaling, a lot of times a cell has to tell another cell it's time to divide before the cell can divide. So these molecules that are sent between cells to allow them to tell each other it's time to divide or it's not time to divide, those are called growth factors. Those are extracellular signals, okay? So when a growth factor binds to the surface protein, this receptor of the growth factor on the cell membrane, it's gonna cause a conformational change of the receptor, which activates the ROS protein. So this ROS protein is called a G protein. We'll talk about G protein in a few weeks, but you just know this is a relay molecule right here. It is activated by a GTP. That's why it's called a G protein, because it's activated by a GTP. So this ROS protein is activated because the growth factor bound to the receptor. The result is this ROS protein is going to activate the next, and the next. We have all three of these protein kinases, and probably actually more than that. These protein kinases can also be called the relay proteins, and they're kinases because they each phosphorylate the next protein. The result is, excuse me, eventually, at the end of this signal transduction pathway, a transcription factor is going to be made, and this transcription factor is actually an activator that produces proteins that stimulate the cell cycle. So when a growth factor is received by a cell, the end result should be this cell is going to divide. But how is this cell going to divide? Eventually, these transcription factors are going to be made to allow proteins to be made to stimulate the cell cycle. So it's always a chain of events. This happens, this happens, this happens. Anyway, so this is what happens in the cell when the ROS protein is not damaged. If you were to have a ROS protein that's mutated, there's a possibility that the ROS protein could be activated even without the growth factors. You see, this GTP is attached to the ROS protein, activates it, even when there's not a growth factor. That means the cell's not supposed to be dividing. Now, the cells should know that it's not supposed to be dividing, but that's not happening because our ROS protein is mutated. So now, as you can see, it's going through this entire pathway that's just as the one on the top, and it's causing an overexpression of the proteins that stimulate the cell cycle. Now this cell is dividing, and there's nothing that can stop it. Well, in your own body, at least, that there's not a whole lot that it can be stopped, unless you do chemo, but that's a different story. So the ROS protein is a very, very important, example for you to understand how protein works. Another example is, earlier we talked about tumor suppressor gene. An example of a tumor suppressor gene is called P53. So this P53 has shown up quite a few times on the AP test. So pay attention to how P53 works. This is what the P53 tumor suppressor gene usually does, okay? The first thing is that it codes for, it activates P21, there's so many P's, P21, which codes for P21 protein. So this is a P21 gene. And this binds to cyclin-dependent kinases. If you can't remember this, as cyclin-dependent kinases is also called CDK. If you remember way back when we talked about the cell cycle, CDK is something that controls the cell cycle, whether the cell cycle is supposed to move on from G1 to, S and G2 and the M. Are we supposed to go in through this cell cycle? The concentration of the cyclin CDK is going to control the cell cycle and allows the cell cycle to go or stop. And what P53 or P51 actually does is that it allows cell cycle to pause when it's not supposed to go and repair damaged DNA, right? So that's what normal P53 and P21 does. P53 also activates miRNA. Remember, miRNA is the police that chop up the foreign DNA, or when there's problems with the DNA. So it inhibits the cell cycle as well, but in a different way compared to the P21. P53 can also activate suicide genes responsible for apoptosis. So remember, when we talk about apoptosis, when there are damages within the cell, apoptosis is supposed to happen, and P53 helps with that process. So if you think about it, if P53, this tumor suppressor gene, is damaged to the point that it's no longer functional, or you have a decreased amount of P53 proteins made, or the tumor suppressor proteins made, all three of these processes and many more processes are going to be stopped. And the problem with that is now your cell cycle is just going to continue all happening. The cell is going to divide continuously, even when there's a DNA damage. And again, cancer is caused when there's an accumulation of a lot of mutations, right? And the only way of accumulating a lot of mutations is when your cell is damaged, and then but they're still dividing, even though it's supposed to stop or go through apoptosis. So the suppression of the cell cycle can be important in the case of damage. What is this? Suppression of the cell cycle can be important in the case of damage to a cell's DNA. P53 prevents a cell from passing on mutations due to DNA damage. So P53 prevents tumor suppression, prevents cancer by stopping the cell cycle when there's DNA damage. But if you were to have a mutation, that ain't happening. More than 50% of human cancer is caused by a damage in the p53 gene. That's why this is important. All right, so if we were to visualize this process, how exactly does p53 works? On the top picture, excuse me, we can see how p53 normally works, okay? So for example, if there's UV light that's causing damage in the DNA, which happens all the time, right? It happens to you, I'm not saying it's on a daily basis, but your DNA, there's constantly damages, and most of the time your cell fixes it. So if there's a DNA damage, this DNA damage in the genome is going to cause a protein kinases, this chain of events happening within your cells, which activates the p53. And the result is this p53 protein is going to actually increase the amount of transcription of this protein that inhibits the cell cycle. So this is a whole chain of events, right? Damage, which activates p53, which activates transcription of a protein that inhibits the cell cycle. However, when there's a mutation, even when there's a damage in the genome, this p53 protein could be defective, or it cannot become a transcription factor. So then the result is we cannot produce any more of these inhibitory protein for the cell cycle. So now our cell cycle can just go on and on and on, even when it's supposed to stop. Now, there's cancer. So because mutations happen all the time, right? So if you just think about it, with people aging, more and more mutations will happen within the body. So normally, older people will be more prone to cancer. You know, let's say two people, the one younger, one older, they all have the same susceptibility to cancer. They all have some potentially cancer-causing genes, but the older person is more likely to actually get the cancer. So this is showing you that cancer is an accumulation process. When there's only a little bit of mutation, that's fine. A little bit more, that's still fine, right? Activation of ROS oncogene. So now, but if we have all of these possibilities, all of these problems messing up with the cell cycle at the same time, then this is really going to turn into, these cells are really going to become cancerous. This is just showing you for breast cancer, there are different types of breast cancer, and as you can see, there are many genes that could cause cancer, but the more of the oncogenes that you have that are actually cancer causing, the more likely that you will actually get the cancer. But it really all depends. So it's complicated. It's most likely not just one factor that will cause cancer, right? So if you were to read an article, big takeaway right here, okay? You're going to get a lot of news, and a lot of the news that are shown to you from the media or wherever, or even on scientific papers, they're real news. They're not fake news. The information is real. But you really, as a science person, now that you've taken AP Biology, you need to look at everything with your own judgment. So if you think about it, if there's could be a title of a paper that says, not of a paper, of an article that says bacon causes cancer, right? If you just look at this title, this is just wrong. The reason for that is because, okay, bacon might put certain people more at risk for cancer, but bacon will never be the only factor that causes cancer, right? So you need to really think about how science works, how biology works. There has to be, things that happen within an organism, and, you will get certain mutations or certain genes from your parents that could already be damaged. That's going to make you more likely to have cancer. And if you have other environmental factors combined with the genetic factors, now this person might actually have cancer. But it really all depends. There's not a single factor, usually, that will directly lead you to cancer. That's why, cancers are so complicated and it's so hard to treat. There's so many factors involved in it. So individuals can inherit oncogenes or mutant alleles of tumor suppressor genes, right? That makes sense because we can inherit a lot of things, everything. DNA methylation and histone protein patterns, as well as miRNA mutations, can also cause cancer, right? So if we're not destroying certain copies of the mRNA when we're supposed to, that could cause cancer. And DNA methylation and histone protein pattern, that's related to gene expression. And if you have unregulated gene expression, then cancer is possible. Okay. So this right here, what is this? So it's just an example. Inherited mutations in this type of APC, a tumor suppressor gene, can cause colorectal cancer. And then here's our two different types, BRCA1 and BRCA2. These are related to breast cancer. So you might see these APC, BRCA, in an AP question again, but they'll always give you the background. Your job is to understand how these genes, well, not exactly how these genes work, but how cancer works, how gene regulation works, most importantly, so that when you read the question, you understand what the question is talking about so that you can get it right. We're almost done. Okay. DNA breakage can also contribute to cancer, right? That's the type of mutation. Mutations causes cancers. DNA breakage can be caused by many environmental factors, such as some big ones: ultraviolet radiation from the sun. There could be chemicals found in cigarette smoke. There could be viruses that can cause cancers, because viruses can introduce new genes or new mutations that can turn a proto-oncogene into an oncogene. So we're done.